Very good morning, all of you. So I'll be talking about decoding spatial selectivity in chaperone mediated autophagy. A lot of keywords which some of you might not be understanding, but I'll keep it a very, very layman language term. The very first picture, since we have this Diwali week, all of you have done this Diwali, right? So who's the cleanup crew? In our cells also, when we have a lot of junks accumulated, what is the cleanup mechanism there? How do cells clean the houses? Is there some vacuum cleaner, some garbage truck? The answer is yes. The process, some of you might not know, is called autophagy. So when we have garbage trucks accumulating all sort of junks, it will be beneficial if we have certain recyclable, certain non-recyclable, right? Similarly, here in our cells, we need to segregate them into different categories, depending upon what type of cargo we need to degrade. So is it mitochondria, proteins, lipid droplets? What kind of cargo we need to degrade? That's called selective autophagy. One such selective autophagy is chaperone-mediated autophagy. That is what my lab works on. So basically, it's an autophagic mechanism, the degradation mechanism, which degrades protein-containing K4Q type motifs. So if a protein has this tag, they are prone to degrade via this mechanism. The mechanism is this K4Q motif binds to a chaperone, which is HSC70. Together, they form a complex, goes to something called lysosomal associated membrane protein that's inside the lysosome, integral membrane protein. It multimerizes, this is multimerization complex. It forms a translocation complex wherein the substrate enters into the lysosome and is degraded. So this is a seminal process that happens. Now there are two key players here. The first one is this LAMP2A. So LAMP2A is an integral membrane protein. It needs to multimerize to form translocation complex wherein degradation further can happen. So this step is very, very selective. And the second step is this HSC70, which recognizes only these motifs. Sequence-wise, we know the motif has to be K4Q, but structural-wise, we don't know. What are the structural preferences? What are the binding sites? Nothing is known. So these two are the key questions that I would like to answer through my research in this talk. So the basic question that we answer is how spatial alterations influenced by electrostatic environment, conformational dynamics, and binding landscape shape CMA functionality. So to this end, to answer the first question about the LAMP protein that I mentioned, what is so special in LAMP that dictates selectivity? So this is the domain organization of the LAMP. It consists of transmembrane part and C-terminal part. This one here. So this, if you see here, the juxtamembrane residue, which is KHH in the wild-type LAMP2A, seminal paper by Dr. Anna Maria Carvio has shown that if you mutate this to alanine, 4 alanines, the functionality is lost. And this made us curious to know what is governing the functionality. So we replicated the same system in silico using MD simulations. We constructed this and made the mutant, AAA, and tried to see what happens in the functional and the non-functional. That means the monomeric as well as the multimeric form. What are the changes that happens wild type versus mutant? We also varied the charges of this since KHHH, which is charged, all the 4 charges. But it also depends upon the local environment. So we varied different charges, keeping K as 1 in all of them, H as 0 in the first case, the first H as 1, the second H as +1, the third H as +1, all the H as +1 charge, and then we have the inactive form, which is AAA. We try to see what are the differences which are governing the functionality. In total, we have 30 microsecond of all atom MD simulation data. So first, we created a mammalian lysosomal membrane, which computationally is quite challenging in the sense it's a heterogeneous complex with a lot of molecules. And, but we wanted to keep it as close as possible to the experimental conditions. So that's how we modeled this. And then we inserted the 3 trimeric assemblies as well as the monomeric assemblies. And this was very interesting to see. This juxtamembrane residue, KHH, on mutation forms alpha helical. You can see the red dots form alpha helical segment, and that alpha helical segment gets embedded inside the membrane. So it was very interesting to see the one which was at the periphery got embedded inside the membrane. So this was one of the key factors which could have led to the loss of the functionality. So probably we think that these residues might be playing an important role in recognizing the substrate. Since now they are buried, there is no functionality left. So this is just the 3 replicas and all the replicas, saying that statistically also we observed in all the simulation runs, this region forms alpha helical, and alpha helical gets embedded inside the membrane. Moving on, we were interested in looking at what are the structural changes this embedding has led to. So for that, we have measured the tilt angles. The tilt angles in all the systems, if you see, alanine, 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 which is the orange one, is more towards the higher end, which means the tilt is much more in the mutant part. Because of this tilting, even the membrane thickness, this is the membrane thickness, we observe substantial change in the membrane thickness compared to the wild type and its variants. And this is local density map, membrane thickness. Also here, we see that alanine has higher local membrane density. So to conclude this slide, we can say that mutation causes increase in tilting along with increase in membrane thickness. Further, so this was just the tilting and the thickness. We were also interested in looking at how the lipid environment is changing, because lipid environment is a very important factor in governing the functionality. Only certain lipid microenvironments are known for function, but what is that microenvironment is we don't know. So we went ahead to see how many contacts of this protein with lipids, which are negatively charged lipids, does it make. So here we can see substantial reduction in the mutant. The mutation led to substantial decrease in the contact with negatively charged lipids. That's also what we expect, because KHH is a positive charge. So this is preferential clustering of the positively charged lipids. This is all diminished here. Then we were interested in looking at what happens to the trimeric assembly, which is a functional state. The trimeric assembly was also perturbed in a large way. The trimers are now destabilized. They are not coming together in the same way as in the wild type. So this is a triangular area we calculated among all the trimeric forms. And we found the area is very, very high in all the mutants compared to the wild type and its variants. And to be more specific, we have collected information about the residue-wise information. And to the residue level, the cytosolic end is perturbed in a larger way compared to the lumen end. Lumen, we still have contacts, but cytosolic, all the contacts were lost. So to summarize, we have created and compared the mutant versus wild type, and we have found that membrane embedding happens in the mutation, and this leads to enhanced tilting and loss of trimeric assembly. And this is recently on the Diwali day, it got accepted in JPCB, and this is accepted front cover also in JPCB. And moving on to the second part, HSC 70. HSC 70, what is the structure selectivity relationship? This is a protein, and we have evidences that it binds to this part. So we have taken this. Using computational modeling approaches, we figured out AlphaFold 3, that there were 3 binding sites. So there were 15 proteins, which we simulated for 1 microsecond each, leading to about 15 microsecond of data. So when we simulate only the motif, which is known to bind, the K4Q motif, the tag, which I was telling you in the first slide, the 3 binding sites were this, but when we subjected to MD simulation, all the binding was lost. So there was no consistent binding site observed. So we were puzzled. Then we thought, why not just extend this interaction? So instead of taking just the K4Q motif, we extended the motifs so that it encompasses all the structural components. So if it's alpha helix, we have taken the entire alpha helix instead of just the K4Q segment. And we found that the binding was now selective. This was very important finding. All of the 15 residues, if we again subjected it to 1 microsecond of data, which amounts to 30 microseconds of data again, we found that alpha 1, alpha 2, alpha 3 were the binding site, and this site is basically hydrophobic in nature. But if you look at the orientation, all were promiscuous. So the question of selectivity is there, but selectivity in the binding site, but not in the orientation. So the road ahead is to again look into what is the orientation which leads to selectivity. To conclude, we have done 2 studies, HSC70, conformational preferences for binding site, and for LAMP2A, what are the structural determinants regulating LAMP2A multimerization. So with this, I'd like to acknowledge Shiv Nadar Institution of Eminence, Shiv Nadar Foundation, Magus 2, which is the HPC facility at Shiv Nadar University, my group, and the first one is Ishwar. He did most of the LAMP work. The second is a PhD student, David. He did most of the HSC70 work. And the others are UGs and PG students. And also I'd like to thank DST Inspire for the grant that I'm using. And thank you everyone for your attention.
Thank you. Questions?
Questions?
Hey, nice talk. I wanted to ask regarding the LAMP part that you presented. You showed that there was a formation of a multimer there, but and you mentioned about certain lipids also. There were a lot of lipids in the lysosomal membrane that you also recapitulated in your simulation data. Did you see a specific lipid that was associated among all of them that was enabling the clustering?
We did see negatively charged lipid, this one. And this was very interesting to observe that this negative charge, which are PI and PS lipids, so specifically SAPI, SDPI, and PS lipids, SAPS and SDPS, along with BMGP. The common thing among all of these lipids were that they are all negatively charged. So these lipids were preferentially associated with this LAMP multimer. And probably this lipid microenvironment is regulating the functionality, which was absent in the mutant. So yeah, this was very interesting thing which we observed and which you also pointed out.
Following up, do you also think this would be really important for fusion process and macro autophagy rather than
Yes.
here?
Yes.
I mean, the clustering of LAMP, has it been shown to be involved in fusion process with autophagosomes?
Not LAMP2A, LAMP2B is involved in fusion processes.
That also forms clusters or these?
That
multimers?
probably we need to check. They form microclusters, but what type of microclusters is not known, not studied yet. But LAMP2B is involved. That's another side project we are working on. So yeah, not LAMP2A.
Thank you.
Yeah, thanks.
The attachment of the KFERQ site onto proteins that are destined for autophagy.
Yeah, so the binding site is not known. What is known is just the sequence preference. So this KFERQ type motifs will go and bind. Where it binds, nobody knows. So that was a challenge we took up using computational approach. And we figured out probably it could bind to this site, hydrophobic site here. But there is a selective site which it binds, but it's not selective orientation. And there is a cross-linking experiment, which I have not shown here, which proves that the site is indeed correct, but may not be responsible for selectivity. But it binds to this site. Selectivity probably could be another site. So we are proposing it's a secondary binding site. The primary binding site is something else.
And what puts the KFERQ on the proteins destined for interaction with HSC70?
Most of the cytosolic proteins. So there was a study which says that around 90% of the cytosolic proteins has this intrinsic. Yeah, so they are all destined to go via autophagy, selective autophagy. So mostly the cytosolic proteins are destined to go for this process.
And
So
so
this
they
tag.
accumulate this tag over time as a sort of a function of protein age, do you think, or what's known about?
So they do not accumulate over time. They have it initially, but they can be tuned for autophagy over time by post-translational modifications to make it to this particular type.
Mhm.
So they can be destined over time with aging, but using post-translational modifications. But most of them do have this KFERQ initially. Yeah, that's a very interesting thing to think about.
In interest of time, let's thank